Okay, let's get started. So we will be doing the test one review today. You can find this package on eCentennial. First question here, label the different layers of the fractionated blood in the diagram below. Fractionated means you collect the blood and you spin it down at a centrifuge. So the heaviest stuff sinks to the bottom, and the lighter stuff is going to go on top. So we have this yellow clear portion of the blood. What do we call that?
Plasma protein.
Plasma, that's correct, plasma. So this would be plasma. And what's inside the plasma? Well, it is mostly water with dissolved ions, nutrients, waste, right? And there's also plasma protein. Okay. So plasma makes up what percent of your blood? 55, that's right. So approximately 55% of your total blood volume. Now let's talk about some of these plasma proteins. Okay, so we call them PP, right? Plasma proteins, we have proteins that are essential for the immune system. So immune proteins, right? Such as antibodies, we have things like complement proteins. We learned about that 2 weeks ago. These are part of your innate immunity, right? And do we have another one here? That's a part of the immune system. I think that's the two main ones that we talked about, right? There are clotting proteins. Okay, proteins that are necessary for stopping a bleeding. And anybody have any examples of that?
Fibrinogen.
Fibrinogen, that's right. Fibrinogen, which is inactive, right? And then there's another one as well. Yeah, prothrombin. So these are all plasma proteins. And there is another one, right? There is one that's called albumin, right, which is primarily responsible for maintaining osmotic pressure. Okay. What's the process of clotting called? Coagulation. Okay. I got someone asking that in the chat. Coagulation. All right. Okay. Next we have all these things. The middle layer, right? That's kind of between the plasma and the red cell. That is where you will find the white blood cells and the platelets. So there's this buffy coating, we call it. That's where you will find the white blood cells plus platelets. Right? And they don't make up a lot of volume, like maybe around 1%. And we learn a bunch of white cells, right? Okay. There is the granulocytes. So neutrophil. There is eosinophil. There's basophil. Right? And then there's also mast cells, right, which are related to basophils, right? And the other category is agranulocytes. Anybody have the list for me for agranulocytes?
B cell.
B cells, T cell, yeah. And then what's the other one?
Monocyte.
Monocytes, very good. So monocytes can become macrophage. There's the T cells, which have many different types, as you know, and B cells as well. Okay. So these are cells where you can see the visible granules. These are the cells that the granules are not visible under the microscope. All right, last part here. These are the red blood cell, also called, anybody remember what it's called?
Erythrocytes.
Yes, erythrocytes. That's correct. Sorry, my question was not clear, I guess. In terms of like this portion of the fractionated blood, we have a name for that, right?
Hematocrit.
Yes, thank you. Hematocrit, which makes up about 45%, right, of your total blood volume. So obviously the RBC is going to have no nucleus, right? No nucleus. It contains hemoglobin. The shape we would say it is biconcave, right? Like if you look at it from the side, it's going to be like pinched in like that, right? Biconcave. And that's important for maximizing surface area, right? For gas exchange. Lifespan, how long do they live for? Yeah, about 120 days. And then you would break them down, you will recycle them. I don't know if recycle is the right word. We'll say break down, okay? Break down the old ones, break down at liver and spleen. Okay. It's possible that you don't have enough red cells because you move to a higher altitude, or like your body's undergoing certain changes that cause the RBC to go down. So there is a hormone from the kidney that would boost the RBC production. Does anyone remember the name of that hormone? It's 3 letters, right? We can use the acronym, the short form, instead of the full name.
EPO.
Yeah, EPO, that's right, EPO, which stands for erythropoietin. So EPO from the kidney will increases RBC production. Okay, and of course, like the white blood cells, the platelets, and the red cells, all these blood cells, they are from stem cells in the bone marrow, right? And the process is called hematopoiesis, right? That's the process of creating these blood cells, okay? From the stem cell, of course. I hope that makes sense. Okay. Compare the 2 test tubes shown below. Explain what might have caused the results on the right. The left one is the normal one right here. You can see the plasma. You can see the hematocrit. Although the plasma here looks a little bit more than it normally would have. Doesn't seem like there is too much red cell here. But for our purpose, let's just say this is the normal one. Normal. Can barely read that. What's going on with the right one? The one on the right side. Anybody want to guess what's wrong here?
What do you mean by?
So this is the normal fractionated blood. That's what it's supposed to look like, right? With the plasma on top and the red cell at the bottom. And so if you look at the one on the right, the plasma is not yellow, right? It's red. So does anybody know what might be wrong with it?
It's because they didn't put it in centrifuge to.
That's a good guess, yeah. But they actually did. Okay, this is already centrifuged, already spun down. Any other guess? This question is on the test, right? So I just want to make sure that you know the answer before you take the test. Anybody know or have a guess of what might be wrong? It's something we talked about in class.
Any hint?
Yeah, okay, yeah, Justine got it. It's hemolysis. That's right. Okay, so this is hemolysis. And you get hemolysis for various reasons, right? For example, it could be caused by mismatched blood transfusion, right? Okay. When antibodies bind to antigens on RBC, right? That would lead to a cross reaction, right? Resulting in agglutination and hemolysis, right? So all your red cells basically burst, and the hemoglobin comes out, and that's why the plasma is reddish now, okay, because of the hemoglobin that came out from the red cell, and that's what happens when there's a mismatch of blood transfusion. I mean, that's one cause of hemolysis. There are other reasons too, right? Like internal bleeding, right? That's another possible reason of why this could happen. So we're clear? If you see this picture on the test and I ask you what's wrong with the sample, you will know what to choose.
Yes. Thank you.
Okay, all right. Any other comments before we move on? By the way, I hope you have done your lab quiz yet. I mean, I saw a lot of people have done it, and the results are quite good. But there's still some of you who haven't done it yet, so please make sure you do that before taking the test. Okay. Parents, blood type on the left here, some babies here on the right. Has anyone tried this question already? Okay, why don't you take a moment and try it? Okay, I'll give you like maybe 2 minutes or so. Have a look at it, and then we'll take it up. Remember, when you see something like this, you do not draw the Punnett square, right? You use the 4 rules that I told you about, right? Like AB cannot have O babies, O cannot have AB babies. Right? Remember those 4 rules? Okay, so go ahead, try that, and we will take it up in a bit. And if you have an answer that you would like to share with the class, please do so. Yeah, Prashal, you have one you want to share? So you unmute.
Yes, Professor.
Yeah.
So number one, A negative and O negative. I guess that is baby K A negative.
Baby, which A negative? So baby K?
Yeah.
Okay. And then?
Then AV negative and O negative. Baby H, AV negative.
You talking about the second one?
Last one.
Last one is H, okay.
And then we have AB, A positive and B positive has a baby J, P positive.
Okay.
And then B negative and O positive has baby I, O negative. Oh, what is this? Okay.
Okay. All right. Thank you. Thank you. Justine have another set. I'm just gonna put it here. What? H J I H K, right? Not A, right? Oh, it's the same. It's the same answer as Pushel. Okay. Thank you. Thank you for sharing the answers. I'm just gonna point out something here. For example, right? Like here, baby H cannot actually be their baby, right? Because one of the parents is type O, and type O individuals cannot have an AB child, right? Because in order to have a type AB child, you have to either contribute an A or contribute a B, right? So someone who has type O will not be able to do that, right? So probably not H. But that's okay. It was a good try. Right? Thank you for sharing. Let's try to do it together. Okay. So here, we can take a look at here. This is type O, and so immediately I can reject AB, right? So that's not theirs. They are both negative, so I can reject the positive as well. Is that okay? So we don't know at this point whether it is baby I or baby K, right? We don't have enough information to say it is one over the other. So I'm just gonna put both here. I'm gonna put I and possibly K. We will cross it out later on. Right? Now this is a good one to work with because we have an AB parent, so O is not the baby. We have an O parent, which means AB is not the baby, right? And they're both negative, which means positive is not possible. So baby K is going to be theirs. Does everybody follow so far? If you have a question, yeah. If anyone have a question, please do ask about how I came up with that conclusion. Okay, so K is the only possible baby for the last couple, and that means it cannot be the first one. So I and K are gone. All right. So next we're gonna take a look at here. We only have two more babies up for grabs. Here we have A positive and B positive, and then B negative and O positive. So this gets a little bit tricky, okay, but it's doable. Right? Let's look at the third couple. There's no A, right, in either of the parents, which means it's not possible for them to have an AB baby, right? No one is able to pass on the IA. Therefore, it's impossible to have an AB baby. So J is their child for sure. And this would be H. Okay. So the answer is IHJK. Okay. Can O negative parents could have O positive baby? Which one are we talking about? Or are we just asking in general, Emma? O general. Okay. O negative can have an O positive baby if the other parent is a positive parent, right? So let's say an O negative marry someone who's like A positive, right? So this would be little I, this would be IA little I, for example, and this would be, say, negative, negative, and positive, positive, then it's possible to have an O positive baby, right? If the A positive parent pass on a little I, and then a little I from here, that gives you the O, and then the positive and negative allele for the RH would give you positive, right? Does that answer your question? Okay. All right. Next, we have this thing that we did before in class, but let's just go through it again. A new blood type called fluffy has been discovered in humans. There are 3, sorry, that's a mistake, alleles in the fluffy blood group, F1, F2, and F3. If F1 is dominant over F2 and F3, then F2 is dominant over F3. How many different phenotypes are there in the fluffy blood group? Okay, so let's list the genotype first. Okay, genotype. So what are all the possible combinations if there are 3 alleles that are present, F1, F2, and F3? So what combinations can we have? I'll give you one. For example, we can have 1 and 1, right? So F1 and F1. F1 on one allele, F1 on the other allele. What's another possible combination?
Small I, or big I.
But there's no I here, right? The alleles are like F1, F2, and F3, right? So we can have 2 of the ones. And what other? Yeah, we can have a 1 and a 2. Good. What else? 1 and a 3 from Mohammed. Yep. Okay. Anything else? I mean, it's just taking these 3 things and then combining them, right? So you can have a 1, 1, yeah, a 2, 2, and a 3, 3, and I'm missing one right here. Okay, yeah, I think some of you got it, and a 2, 3, right? That's all. Right? That's all the possible combinations, right? Because humans are diploid, we can only have 2 alleles at a time. Doesn't matter if there are like 4 or 5 alleles out there for a particular gene. We can only have 2 at maximum, right? So that's all the possible combinations. Okay, so now the phenotype, right? Like what do they actually look like, right? Okay, so let's take a look. The question tells us 1 is dominant over 2 and 3. That means 1 will cover up 2 and 3, right? So let's take a look at the first one, right? The first one, 1, 1, right? 1 and 1. What would that look like? 1 and 1, well, there's only one allele here, so we'll call that like fluffy 1, okay? Fluffy 1. Okay. What about 1 and 2? 1 and 2, what would that look like? Is that fluffy 1, fluffy 2, or fluffy 3?
Fluffy 1.
Fluffy 1, that's right. And how do you know that?
Because the F1 is dominant over F2.
Exactly, because F1 is dominant over F2, it means it will cover it up, right? So 1 and 2 will also look like fluffy 1. In fact, 1, 3 will also be fluffy 1, right? Okay. 2, 2. Well, 2, 2 is just going to be fluffy 2, right? Fluffy 2. Do we have another fluffy 2, or that's it?
3.
Yeah, 2, 3, right? Because 2 is dominant over 3. So that
No,
will
we
be
have another fluffy.
yeah,
Like
fluffy
3,
2.
3.
That's right. 3, 3 would be fluffy 3. Okay. I hope that makes sense. And again, if it doesn't make sense, ask. I don't know what you don't understand if you don't ask, right?
So what does it mean by fluffy in this question?
Oh, it's just a name, right? Like, could it be anything? Right? It's like, it could be like RH, right? Okay, so like you call Rh positive or Rh negative, right? So it's just like a theoretical blood type, right? That I made up. Okay, if all three alleles are co-dominant with each other, how many phenotypes are there going to be? Let's do a poll on this one. Let's see if you guys actually understand. Okay, how many different phenotypes will there be? Okay, let's go A, B, C, D, and we'll say 3, 4, 5, 6. Co-dominant means both are expressed, right? Like IA, IB gives you type AB blood. Okay. See that a lot of you are still not quite sure. Okay, so would the genotype change for part B? Are we gonna have a bunch of different genotypes, or can we just reuse the same one?
You can use the same one.
Same one, right? Because it's the same three alleles, so the combinations do not change, right? Okay. So 1, 1, that will be one type, right? 1, 2, that will be another type now, because 1 is no longer dominant over 2, right? So if you have F1 and F2, that means on the surface of your cell, you would have fluffy 1 and fluffy 2 protein, right? So that's one phenotype. Right? 2, 2, right? 2, 2, it would just be F2, F2, right? F1, F3, 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 F2, F3. So there will be a total of 6 possible phenotypes when they're co-dominant. Okay, so this is the same as saying, you know, you have IA and IB. What blood type does that give you? It gives you AB, because A and B are co-dominant. Both of them will be expressed. Okay. Think about that. We had a similar question in the study guide, right? So go through that as well. Okay. Find the genotype of question mark. Again, you can try that if you haven't already. And we'll take it up in a bit. If you have an answer that you would like to share, please do so. Okay, we have B and A. Anyone else? All right, let's take it up. So we will write down the alleles for people that we are certain of, such as O, right? So we know this is little I, little I. This is going to be IA something, IB something, right? Little I here. IA, IB. IB something, IB something. That's all we know to begin with. So one of the little I came from the mother, and then the other little I came from the father, right? So that much we know. Okay. But what about the other half? Well, the IA has to come from one parent and the IB has to come from the other parent, right? But we got to trace back one more generation. You can see in the grandparents level, they are both type B blood. So it's impossible for their son to be type A. For that reason, this must be a type B person. Okay, so this B came from this parent. And on the other hand, running out of colors here, the A must have came from here. So that's going to be your IA. Okay, so the answer is B and A. Okay. Number 6, let's try that. Go ahead. Anyone else? Has been a minute. I'm going to close the poll soon. Okay, thank you. So let's take a look at this. A drop of Sarah's blood was mixed with anti-A antibodies. So basically, if you think about what you saw in the lab, right? So there's basically like, 3 wells here, right? There is anti-A here, anti-B, and then anti-RH. So what happens is, you would put the blood into each of the wells, and each of the wells would have the respective antibodies. And clumping was not seen in any of the tests. So no clumping here, no clumping here, no clumping here. So let's see. The test must have been defective because clumping should have been seen in at least one of the tests. That's not true. Okay. If there's no reaction for any of them, it just means your cell is not expressing any of the antigens, right? And that certainly is a possible outcome. None of the antibodies in Sarah's plasma reacted with the antibodies. When we adding the blood to the wells, we are checking to see if her red blood cells are reacting with the antibodies. We're not trying to see if her antibodies will be reacting with these antibodies, right? Okay, so none of the RBC, right, in Sarah's blood reacted with the antibodies. That would have been a correct statement. So that's not it. Sarah's blood type is O positive. It's not O positive because there's no reaction with RH. Okay, for it to be positive, you need to have a positive reaction. You need to see clumping with the RH antibody. So what does it mean? Well, in fact, it means she is O negative. So she does not have any ABO or RH antigens on her RBC. Right? That's the correct answer. Okay. Questions? Let's try the next one. Safe transfusion. Select all the safe transfusion here. Okay. So, positive cannot be given to negative. That's not safe. AB negative to AB positive. That's safe, right? AB, same blood group. Negative can be given to positive. A positive to AB positive, that is okay as well, right? Look at the transfusion chart, right? Okay. B negative to AB negative, that is okay. O positive, O negative, not okay. Positive cannot be given to negative. So the safe ones are B, C, and D. Okay. You should have a sticky tag next to your monitor or something when you do the quiz, do the test, right? With the transfusion chart on it. All right, next part. Fill in the following chart related to what do we have here? Related to the function and classification. Yes, is there a question? No, just okay. All right, that's fine.
Yeah, I have a question.
Yeah,
So
go ahead.
B, can you please explain me? B minus 2, AB minus 4.
Sure.
Thank you.
Which one? Sorry, the D, did you say? Why is this okay?
D, yeah.
Yeah, okay. So first of all, negative can be given to negative, right? That part is okay. B to AB. Right? So what's on the surface of the RBC for type B? It would just be B, right? What kind of antibodies do we have for someone who's type AB blood? If someone is type AB blood, what kind of antibodies do they have? Is that 11, Emma? Sorry, didn't really show up. Okay, so no antibodies, right? No antibodies for someone who's AB, right? Because you cannot have anti-A, you cannot have anti-B. Otherwise, you would start attacking yourself, right? So because there's no antibody for someone who's AB, it is okay for this transfusion to occur, right? No antibody is going to react against the B on the red cells. Does that answer your question? Got it. Okay. All right. Okay, next one. We have a bunch of cells here. We can classify them. We're going to write some functions related to them. Hopefully it'll make sense. First one, we have the macrophage. Just to refresh your memory, the macrophage differentiates from monocytes. Okay. And we would classify macrophage as a couple things. It could be a phagocyte, meaning it is capable of doing phagocytosis. It is also an antigen, presenting cell, right? And APC. And we can also say it is a agranulocyte. Okay. So there you have it, 3 types of cells. 3 types of classification, rather. Main functions, what do they do? Well, they just perform phagocytosis, right? On anything expressing PAMs. Remember PAMs? The pathogen-associated molecular patterns. Right. So a bacteria might have some PAMs on it, and immediately the macrophage would recognize that as foreign, and it would phagocytize it. It would eat it up. Of course, being an APC, it is able to do antigen processing and antigen presentation. Right, so we chop up a bit of the bacterial antigens, display it on its surface, and then it will present it to the naive T cells, right? And activate them. So that would be like the last point. Activate naive T cell. All right. Neutrophil. We would classify that as a granulocyte. It is also the phagocyte. The main function is just phagocytosis. Okay, not everything that eat things up is going to be able to present them. Only the macrophage can do it, not the neutrophils. Right. Probably worth mentioning that this is the most abundant WBC. Ahmed asked, Lab 1 and Lab 1 quiz are the same? No, not really. Lab 1 is the PowerPoint slides that you have to download and do on your own. That's not worth any marks, right? Lab 1 quiz is the one that you have to do online, right? With like an actual assessment, right? With the multiple choice questions, right? I posted a link for the Lab 1 quiz. Is that clear?
Yes, Professor.
Okay.
So, Professor, I'm done with the quiz, but I have mistaken in one question. Now I don't know which question I have.
Yeah,
done
you're not
wrong.
supposed to know, right? Because the quiz you have 2 attempts, right? So like, you know, if I show you which ones are the wrong one, then when you do it the second time, then people are just gonna fix up the ones that they got wrong, right? So it's meant to be that way. Yeah, I know it's not ideal, but
Okay, Professor. Thank you.
yeah, okay. Mast cells and basophils. The difference between mast cells and basophils is, I mean, they are both they both play a role in inflammation, but the mast cells, these one, they are anchored into tissue. Anchored to tissues. Whereas the basophil, these are in circulation, right? Okay. And we will classify them as granulocytes. Okay. And the function is to initiate inflammation by releasing what chemical? What's the chemical that they release to trigger inflammation?
Histamine.
Yep. Thank you. Histamine. Okay. So the mast cells, mast cell. These are for localized inflammation, whereas the basophil, that's more for systemic inflammation, like the whole body. Next, we have the NK cells, the natural killer cells. The function or the description, classification would be it is part of innate immunity. Right? The function of NK cell is they would recognize and target or attack, you can say, attack infected cells and cancerous cells. Okay. I mean, it's pretty much everything in your notes, right? We're just writing it out. I strongly believe writing these things out helps you remember them a lot better than just reading your notes, right? A lot of people study biology, anatomy, they just read the notes, they just read the PowerPoint slides. That's not too effective. It's better that you write it all out, in my opinion. Helper T cells. Okay, so helper T cells would be part of the cell-mediated immunity, right? Right? It is the adaptive or acquired immunity. What's the function of helper T cell? Anybody remember?
Activate B cell.
That's right, they activate. Thank you. They activate naive B cells. Okay. And in doing so, coordinate immune response. Plasma cells. So instead of being cell-mediated immunity, plasma cells belongs to the B cell branch. And so we call it antibody mediated immunity. And of course, this is also part of adaptive or acquired immunity. The job is to mass produce antibodies. All right. So there you have it. Summary. Let's try a multiple choice question right here. Go for it. Just one correct answer here.
Teacher,
Go back.
please.
You want?
Yes.
Okay. All right. We'll hold on for a second for the poll. Let me know when you're okay. Okay.
Yeah, I'm okay. Thank you, teacher.
Okay. No problem. Let's try the poll question right here. Okay, has been almost 2 minutes. I only have 7 responses, but that's okay. We'll take it up. This is a little bit tricky, right? So basically, you're being asked to compare specific versus non-specific defense. Specific defense is, of course, talking about adaptive immunity, whereas this is talking about innate immunity. So let's take a look. Specific immunity only works only after non-specific. Specific immunity works only after non-specific defenses have failed. That's not true. Okay, it's possible for them to work parallel to each other. Okay, it's not mutually exclusive, right? In fact, they feed back to each other to amplify the effects, right? The antibody that you produce can end up in enhancing phagocytosis, for example, and so they do not work in isolation. Non-specific defenses and specific immunity require a prior exposure to be effective. That is also not true. Okay, you don't have to be exposed before in order to mount an effective response to it. In fact, like that wouldn't even make sense. Like if you get infected by, again, like chickenpox, for example, for the first time, right? How would you be able to fight it off if you never had prior exposure, right? Like that just doesn't make sense, right? You don't need to have prior exposure in order for them to work. However, once you have had prior exposure, the next time you get infected by the same thing, you will be much better at fighting it. Okay, you will be able to get rid of it much faster. Okay, but prior exposure is not a prerequisite for them to work. B cells work in both non-specific defense and specific immunity. That's not true. B cells are only found in specific immunity. T cells, same story, specific immunity only. Specific immunity changes throughout a person's lifetime, but non-specific defense does not. That's correct. Okay, that is true. All right. Oh, my monitor just died on me. I'm not seeing anything. Hold on. Give me 1 second. Okay, let's take a break. Let's see if I can fix the monitor. 5 minutes, okay, and then we'll resume. I changed the monitor. It should be good for now. Okay. Next one, number 10. Which of the following statements describes a form of acquired immunity? So acquired immunity means that you have to make the antibodies, and go through the T cell selection and all that stuff. Okay, so let's go through some of them. A RhoGAM injection is an example of artificial active immunity. That's not true. Okay, RhoGAM is for prevention of sensitization. It's not considered as a form of active immunity because number one, you didn't produce the antibodies, and number two, it's not used for protecting against a pathogen, right? Number two, a RhoGAM injection is an example of natural. That's not true either. Okay, it's not natural. Anytime you inject anything into the body, it is not considered as natural. Nursing an infant to provide the antibodies in breast milk is an example of natural active immunity. That one is also not correct because it is not active, right? The antibodies is given to the infant, and so that would be considered as passive. For it to be active, you have to be the one who's making the antibodies. Catching COVID-19 and recovering from it should give you natural active immunity. That's the correct one. Okay, you naturally caught a disease, you recover from it by making your own antibody, right? That's natural active. Receiving an mRNA COVID vaccine is an example of artificial passive. It should be artificial active. Okay, so the correct answer is D. Okay. Name the cell. Uses porphyrin to put holes in infected cells. That would be cytotoxic T cell. Okay. Also known as killer T cell, right? Okay. Releases histamine to initiate local inflammatory response. That would be mast cells, right? Local is the key word here. A type of antigen presenting cell that activates naive T cell, that would be macrophage. Kills cancerous cells, that would be NK cells. Forms blood clot, that would be platelets. Most abundant full form of NK cell is natural killer cell. Most abundant WBC, that would be neutrophils. Okay. Give examples of each of the following. Formed elements. What are some examples of formed element? Anybody? Formed elements. What are they? Okay, so that would be the RBC, WBC, and platelets. Right. Of course, this is same as erythrocyte. Someone mentioned that before. This is leukocyte. And this is thrombocyte. Granulocytes, we have neutrophils. We have basophil. And we have eosinophil. Two types of lymphocytes that we talk about, T lymphocytes or T cells, and B lymphocytes or B cells. Coagulation proteins, we only learned about two. There's prothrombin and fibrinogen. Autoimmune diseases, it's probably in the lecture recording. Does anybody know any autoimmune diseases? That's when your body attacks your own cells, right? HIV, it's not an autoimmune disease. HIV is immunocompromised, but good try. So like multiple sclerosis, right? Yeah, Graves' disease, that's another one that was talked about in the lecture. When your white cells attacks your thyroid. Yeah, lupus is another one, Ryan, very good. Not anemia. Anemia is caused by either not having enough red cells or not having enough hemoglobin. Physical barriers, intact skin, right, normal flora, and a bunch of other ones, right? Mucous membrane. Immunodeficiency is when you have a compromised immune system or you don't have an immune system. So we have HIV. Well, actually, that's the virus. HIV causes AIDS, right? And the other one is SCID, when you are born without an immune system. Yeah, Mavis says SCID, very good. Lymphatic organs, spleen, tonsils, thymus. All these are part of your lymphatic organs, appendix. An example of artificial passive immunity. Anyone? Looking for a single word here. What is an example of artificial passive immunity? What do you receive?
Vaccine.
Vaccine would be artificial active immunity because you still have to make the antibodies. Serum, remember, guys? We talked about serum, right? Yeah. Serum, it's purified antibody. Remember the example where you get bitten by a snake, right? You need the anti-venom, right? It's in the form of serum. Natural passive immunity would be like breastfeeding. Characteristics of inflammation, we have redness, swelling, pain, and warmth. Right, the swelling and the pain is caused by increased permeability of the blood vessel, and the redness and the warmth is caused by vasodilation. I think we did these questions already in the during the polls for those lecture. I'm just going to circle the answers for you. You can revisit them later. Okay, I'm pretty sure we did these already. Okay, that's it for lecture 2 and 3, right? And then this is lecture 4. What is the expiratory reserve volume of this person? Okay, so which of these letters correspond to the expiratory reserve volume? Actually, you know what? None of them corresponds to the expiratory reserve volume. It is right here. That's the expiratory reserve volume. That's how much air you can breathe out after your regular exhalation, right? And so if we try to calculate how much this is, we'll say this is about 2,400-ish. This would be about 1,400-ish. And then we subtract them, let's say about 1,000 milliliter. The graph on the test would be much clearer. Right? There's a bit of approximation here, but the one on the test will be very clear. What is the label B? Anybody know what that's called? B is from here to here. What is that? Anyone?
Inspiratory reserve volume.
Good try. Good try. Yeah. Mavis says a VC. Yeah, that would be correct. What does VC stand for? Remind me, please.
Vital capacity.
Yes, that is a vital capacity. okay, which equals to IRV plus tidal volume plus ERV. We did a math question related to that in class, right? So make sure you review that on your own. Describe the breathing pattern of this person. I'm just going to verbally tell you. So the person is inhaling and exhaling regularly. And then there is maximal exhalation. The person breathes out as much as they can, okay, before breathing in again. And then normal breathing, and then maximal inhalation. Right? This part here, that's the inspiratory reserve volume, right? And then returning to normal, and then inhale maximally again. And then from the highest point, they would breathe out all the air that they could, right? And then go back to normal breathing. Describe the volume label as A. I know that there is another A here. So we're talking about this A here. That would be the residual volume. We talked a lot about the residual volume last time, right? Remember, it's high in carbon dioxide. It is not useful for gas exchange. It does not participate in gas exchange. And we don't want to have a lot of residual volume. Okay. That's that. Draw a flowchart to illustrate inhalation and exhalation. Sure, we can do that. Okay, so let's do inhalation. Right, that's for breathing in, right? So in order to breathe in, right, two things must happen. Number one, diaphragm contracts, moves down, and flattens. Simultaneously, at the same time as this is happening, the rib cage moves up and out. So these two things happen simultaneously together. And then, because of these actions, pull the lungs with them, that would increase lung volume, which be equals to decrease lung pressure. Okay, so this has a name, right? This is called Boyle's law. Oops. So what happens next is when pressure in the lungs is less than pressure in the atmosphere, right? We recreate a pressure gradient. Right? Then air rushes in. Oh my goodness, why is the color not changing? Air rushes into lungs. So that's how you inhale. Exhalation is the other way around, right? You should be able to do that on your own. The diaphragm relax and go back up, dome shape. Rib cage comes down and in. That compresses the lung volume, which increases the lung pressure. When the pressure inside the lungs is greater than the atmospheric pressure, air rushes out. Okay. Just a couple more questions here. During swallowing, what happens? The larynx moves up against the epiglottis. Oh, that's the correct one. Okay, it's I told you in class, right? Like if you put your hand in the throat here and you swallow, like you can feel it goes up, right? That's the larynx pushing against the epiglottis, sealing the airway, so the food does not go down the wrong tube. Surfactant, we talked a great deal about surfactant last time. It is the oily substance that is secreted by septal cells. So what does it do? Does it create a moist surface for gas exchange? No, that's the job of water, right? Does it increase surface tension to allow exhalation? No, it actually breaks surface tension. By breaking the surface tension, it increases the compliance of the lungs, which would allow for inhalation, right? So basically it increases compliance, right? Increases the expandability of the lung. Okay, what about the last one? The urge to breathe. The urge to breathe is driven by, let's just do one last poll here. Go ahead, tell me what it is. Yeah, most of you got it. The urge to breathe is driven by an increase in carbon dioxide, and this is detected by the respiratory center located in the medulla oblongata. All right, the part of the brainstem, right? So that's it. There's your review for the test. Anything else you guys want to ask? Any particular concept that you're not clear about that you want to go through before taking the test on Thursday? Anything at all? Well, if you think of anything,
Professor,
yeah, go ahead, please.
is this record will be available?
Yes, I will post
At
it
the
after
class?
class. Yeah. Mhm.
Thank you.
So if you think of anything else, feel free to email me, and you know, we can talk about it. No class next week. Next week is engagement week, and we'll resume on the Thursday afterwards. Okay. Ahmed asked, highest pressure is in vein. No, highest pressure is in the arteries. Okay, arteries have highest pressure. Veins has the lowest pressure. All right, thank you for coming. Good luck with the test. Make sure you study before you take the test, obviously. I will see you in 2 weeks then. Okay, have a good one, guys.
Professor,
Yeah.
Professor, I'm messaging something. Can you please hold a couple of seconds?
Yeah, sure. Go ahead.